Okay. So this roundtable, we have an hour. So Christy, if you can alert me when we are close to done. So we decided for the question and answer, we are going to open it up to the floor. But what we decided that would be much more efficient was to take questions preemptively. So we did that on social media, and we had over 50, maybe almost 60 questions that we received from the community, which was awesome. And the good news is many of them were the same question, asked in different ways. So rather than waste 50 minutes of time of the same question asked in different ways, when this is an amazing opportunity to have these key opinion leaders sitting at the same table. You'll never get this. This is unbelievable. I thought it would be best to use our time wisely and take those questions and put them in an order of preference in which the audience asked those questions. So if it was asked more than once, I put those as priority questions and tried to get everyone's questions on the list. So if we run out of time for those questions, then we'll also take some questions from the audience. And I do think we'll have plenty of time. One thing I do want to do is introduce you guys, but also let you know that because we only have 15 minutes and there are many, many questions, I sent them in advance so that they had 1 to 2 minutes to answer their question. So we want to make this very efficient. But I also want to just emphasize the fact that we have many people sitting up here that are all working on different strategies for gene therapy or gene editing. And because of that, we are in a very pre-competitive environment. So we're not here to compete. We're not here to say mine's better than yours. This group is really good about understanding that all we want is what's best for Angelman syndrome and what's best for our children. And it doesn't matter who does it and who does it first, because some people might benefit from one where other children benefit from another. And that's what you have to keep in mind. So there's not going to be comparison questions. There's not going to be who's better and who's worse. There are going to be questions about toxicity and things that you really care about, and different people's methods. But keep that in mind. And none of the people sitting up here are a pharmaceutical company or a biotech or a biotherapeutic company. These are the researchers who are working in Angelman syndrome every day. And so their agenda is not for that specific company necessarily. So just keep that in mind. So they can answer questions to the best of their abilities in that regard, but this is not the FDA, so they really can't answer some of these questions. They can maybe predict. This is not the pharmaceutical company. They're not making the price tags for things. So those are not questions that they can actually answer. So they may dodge some of those, and I'm still going to ask them, but they may dodge them, and that's okay, because we don't want them to answer questions they're not comfortable answering. So let me just introduce Dr. Ed Weeber, who you just heard from the University of South Florida, and Dr. Kevin Nash from the University of South Florida as well. These are two members of our FIRE team. So we're obviously so grateful that they're here, and they're going to be talking about a gene replacement and or gene secreted protein replacement therapy. It's very hard for people to understand the difference, so you guys will explain that. Dr. Scott Dindot from Texas A&M, also a member of our FIRE team, who really is a world leader in not only making animal models, he did make the pig and he did make some of the mouse models of Angelman syndrome, but also of the antisense transcript and understanding the paternal allele and why it's, or how it's off, and potentially how we can best turn that on. Dr. David Segal from the University of California at Davis, who is working very, very robustly on the zinc finger or artificial transcription factor approach, and also has some involvement in the hematopoietic stem cell gene therapy approach. Dr. Beaudet from Baylor College of Medicine, who really is one of the founding fathers of Angelman syndrome. We're so lucky that he's here today. Not only was he intimately involved in discovering the gene EB3A, or we wouldn't be having this panel if it weren't for him, thank you very much, but he also has been really a leader in activating the paternal allele and really trying to push that strategy forward as what he feels is going to be very beneficial for our kids. Dr. Ashley Winslow, she is actually the head of translational research between the gene therapy program and the Orphan Disease Center at the University of Pennsylvania. So she is our connection and our contact in terms of translation to the foundation of what's happening on a regular basis at the gene therapy program at Penn. So she's going to be able to answer a lot of those questions. And then obviously Dr. Jim Wilson, our keynote speaker, who is a huge pioneer in virus delivery, viral vector, and gene therapy. So I'm going to start with the first question. So this was a very commonly asked question for the panel, so I'm going to go through each of you to try to answer this as briefly as you can. But for each member, that's worked toward a therapeutic for Angelman syndrome. What do you feel it is about Angelman syndrome that makes it so appealing to those of you developing gene therapy strategies and to the various biotherapeutic companies that are currently in this space? Dr. Weeber.
Actually, I was drafted into doing Angelman syndrome research by you guys, so...
I'm pretty sure that was by Paula Evans. Let's just
well, it was by Paula Evans, and there was definitely a number of you. So for the gene therapy, you have to remember back when I did this with Jen Daly, and Jen Daly did all the work, but we were simply trying to answer the question of can we put the protein back in an adult mouse that had Angelman syndrome and try to reverse and rescue those symptoms, those phenotypes. And so it kind of bloomed from there. So, for me, it was the success of that first experiment.
Well, I kind of got roped in by this guy, so that's how I ended up here. But we collaborated together because he wanted to do gene therapy. I was in the gene therapy field, so we started working together. And I like Angelman syndrome because it's a monogenic disorder, so it has really good potential for a gene therapy approach.
So I got roped in by this guy. No, I think that one thing that is very peculiar about Angelman syndrome and this gene EB3A is the fact that one allele is turned off and the other one is on, and so it provides a very unique opportunity to develop strategies to target that allele that's been turned off, and it's a naturally occurring phenomena. And then based off the work that we've done and that other groups have done, the mechanism by which that occurs is very different. And so, and I think that underscores the ability of people to be able to turn it back on. So it's a very different mechanism that it uses to turn that allele off. And so, as you've heard, people have been able to exploit that to reactivate it. And so I think that's something that's very unique about this condition.
So I think between the three of them, they must have stole my answer there somewhere. But I would just say that, in a lot of the 6,000 or 7,000 monogenic disorders that are out there, you have two bad copies of the gene. Usually you have two bad copies, and then when you have two bad copies of the gene, there's not a lot of choices except for a gene replacement kind of therapy that we heard about earlier. And that's still a candidate for this disease as well. But like Scott was saying, in Angelman, there's a very unusual opportunity that there is a healthy gene that's right there in the body with its normal regulation and control and isoforms and everything else that the gene has to have. And with the types of tools that I've been working with, it was an unusual opportunity to apply those particular tools. It seemed like a good marriage between the types of tools that we were working with and a disease that could actually help people. But I know I don't have much time, but I would just say, I think it was the science that really brought me into this, but it was this Angelman community that really made me want to stay.
One of the very attractive things to me is that I deal with a lot of different children with genetic disorders, and in many, many of the disorders, the brain never forms properly. And to think about a cure is really, really almost untenable if the brain never formed properly. In Angelman syndrome, in the mouse and in the human, all the evidence is that all the right nerve cells are there, they're connected in appropriate ways, and so I think that's one big attraction. There's just many disorders where you couldn't really entertain a cure, I don't think. The other is that there is this alternative copy of the gene, and it's a little even better than that. The paternal and maternal copy of the gene both seem to be starting to make the product. They both activate transcription. But on the paternal chromosome, it kind of runs into the antisense in a collision, and so it isn't a productive effort. But if you take away the antisense, the suggestion is that maybe the paternal gene will behave exactly like a maternal gene will behave, because that's what it's doing anyway, except it's got this obstruction. And so, as was already mentioned, the fact that you should get all the right isoforms in all the right cells and these kinds of things.
So I think everyone stole my answer. Art took the last remaining one I had. But I think just the myriad of different options you have for therapeutics to see, having worked in the orphan disease space, to see a rare disorder being addressed by so much research, so many molecular therapeutic options is just really exciting, and I think that's one of the reasons that we got excited to join this effort. There's a lot of potential. A lot of the puzzle pieces have lined up for the reasons that have already been spoken about. I think there's a lot of potential in this space.
At the Orphan Disease Center, we're approached frequently about engaging in new programs, and we actually have some discipline around the decision, so it may not be as a and it really was technical feasibility. Do we think we can pull this off? We think if we learn more that we can. Is there a good animal model? It's not great, but it's a lot better than most, actually, in most diseases. And is there potential reversibility of the phenotype? Speaking to Art, is the anatomy intact? But we were convinced that there was a potential for reversibility. But I got to tell you what struck me was when we engaged in our diligence, that fast brought the community together, just like that, in a way in which the conversations were critical, challenging, civilized, and got us to the point where we got the information that we need. And that carried the day, because I can tell you many other orphan disease communities aren't as cohesive as this one. And if they're not, it's a nightmare. And congratulations to you all for bringing the community to where it is. But that really carried the day.
We're relentless. Okay, the next question. For each gene therapy strategy that is currently in development, where does each specific strategy stand that you're working on, and what is your anticipated timeline for it to be phase 1 ready? So some of you already kind of answered this, but I'm going to put a little bit more pressure on you because I know you can't say something, but maybe you will. So for Dr. Wilson, for your gene replacement therapy that you already discussed, your 18-month kind of predictability, but it depends on how long it takes you to get to that 18-month mark. What is your prediction to get to that 18-month start time?
You know, I've sat on these panels for 20 years, and it's hard to say. I mean, I usually am too conservative, so. But let me put it this way: that if we take the existing components that we have assembled in other programs, which we've assembled to vet them in animal models, if that looks sufficiently encouraging, and there doesn't seem to be something over the horizon, then you know, we would be at that point of a pre-IND meeting. We say within 12 months. But for us, from the time that we have the pre-IND meeting to the time we're first in human is about 13 months. Because we've done this, well, look, we got to get there, but this is the power of the platform that, you know, we work with 5 CMOs, and this is turnkey. But we got to, 1 year from now, we want to reconvene and see if we have it.
Wonderful. Dr. Segal.
Well, the work that we've been doing up to this point is kind of certainly in the preclinical testing phase. And we've been testing a lot of different kinds of routes of administration and some fundamental properties of the approach. But we imagine that work will be drawing towards an end, and that will be moving very quickly into doing behavioral testing in mice, and hopefully also doing some early investigations in large animal models as well. And we expect to be at that phase by the middle of 2018. Now, after that, we would need to partner probably with another entity that maybe like a pharma company or something like that, that would be interested to carry this. into more preclinical trials looking at safety and efficacy in a more focused way and a highly regulated way. And that could take a year or two after that.
Dr. Beaudet?
Well, I should be clear that I have no say whatsoever, but Ionis controls all of this kind of thinking in my situation. Although when we completed the mouse work, they said they very much wanted what they referred to as a humanized mouse. They wanted a segment of the human antisense sequence put into a mouse so they could use oligos that were directed against human DNA, but they could do whole animal experiments. It turns out they wanted a huge piece of DNA to be put in, which is technically very, very difficult to put it in a particular location, in the right orientation. And there's been lots of delays, and I finally started putting in smaller pieces. Now we have some smaller pieces of human DNA that are doing exactly what we would like them to do. I think maybe the Ionis is becoming a little more flexible about that. They seemed to indicate when we first started this that they thought it would be at least a year from the time one had this ideal humanized mouse to do the subsequent studies to get to a phase 1 trial.
Art, can I ask you a question about that? So I wasn't here earlier to participate in the discussion, but I'm familiar with Ionis work through the work that we've done with Biogen for SMA, and it's a platform. It's a really incredible platform that they have experience with the platform as we do in gene therapy. So what we're starting to do in gene therapy is to leverage the platform for many aspects of evaluating the pharmacology and toxicology, and actually do the pharmacology in vitro. So in other words, if you could vet the ASOs in an in vitro model and then leverage the platform of the delivery of the ASO, is there any combination there that would have a path forward?
Yeah, I think a lot of the things, well, of course, these ASOs like the SMA ASO and an Angelman ASO only differ in sequence, and the chemistry is otherwise somewhat similar, and so they have information like distribution in primate brain and so on. I think the platform could speed it up. They have been I think they're concerned that they want to show that the oligo to be used in the human is safe in a living animal and where it's doing its in vivo thing. They're very focused on that, so I can't really speak for them beyond that.
I'm not going to ask Dr. Weeber and Nash because I think Dr. Daly very clearly said end of 2019, so I don't think you probably have a better answer than that, I'm assuming. I obviously can't ask Roche because they made a great announcement today, and I think if I push them on this question, they might leave. So I'm going to leave them out of this question for now because I feel like we got a lot of information from that today. So this is a question for Dr. Weeber and Dr. Beaudet. Can you express your thoughts on the potential, air quotes, therapeutic window? How do you translate the age of a mouse to the age of a human who is obviously far more complex? Do you feel a treatment will benefit all ages? Dr. Weeber, do you want to start?
No. So yeah, how do you translate that? Therapeutic window has come up. I think that, and I'm assuming that Dr. Beaudet will agree that I think traditionally the earlier in life you can treat something, the better the outcome. But we were at the ABOM meeting yesterday, and we saw some interesting data where people are looking at types of ways to measure human learning and memory and cognition, and there's different strategies even in adult AS patients above 18 years of age. So I think it's difficult. We're in completely unknown territory. For the age of the mouse, trying to translate that into a human, we talked a little bit about that earlier, and we do all of our work in adult animals, so I don't know what they translate out to be. Probably in the early 20s, I guess, is what you would consider, but certainly adult animals that we're rescuing. So we don't go in try to rescue very, very young mice. We do it all in the adult, and we've based all of our strategies on that.
Dr. Beaudet?
I don't have anything to add about the age of mice versus humans. There is some data on this. Dr. Ipe Elgersma in Europe has a mouse model where he can turn on the Angelman gene in a mouse that starts off life without expression of the Angelman gene. And he's looked at various windows of when certain skills might be recovered or not recovered. And it varies, and I think there's no question that the earlier, the better. It is encouraging, though, that he suggests that you can correct this LTP thing, which you've seen demonstrated at any age. So it's a little bit, I think maybe the most favorable kind of circumstance would be like hemophilia. Suppose you have a hemophilic boy, and you really protect him from any kind of activity. You don't let him, do anything where he might bleed. And finally, at age 20, there's a cure. He would benefit just as much from that cure at age 20 that he would at 2 days of age. And maybe you can think about the LTP like that. So there's room for some optimism. I could imagine that a 20-year-old Angelman might say a few words. He's not going to all of a sudden have a vocabulary like your vocabulary and my vocabulary. That takes a lot of learning and years of learning. So I think there's room for optimism, but I also think it's clear that the earlier that one could treat, and I think if we had a really definitive treatment, newborn screening would be implemented easily, and most children would get treated under 6 months of age. But all of the people in this room have children who are already 10, 20 years of age. So I would say, I think there would be a lot of benefit in a 2-year-old. I'd be much more concerned about a 12-year-old. In between there, I think it's a little bit hard to know. I think it's hard to know at any of the ages, but anyway, it's a few perspectives.
And along those lines, for Dr. Wilson, because you've, I think, engaged with the FDA more than most people on the panel, would you feel that there is evidence enough or support enough with this day and age of the FDA and the leadership at the FDA to encourage clinical trials in a younger population than the traditional adult starting population? The ultimate being not because young kids deserve it more than older kids, of course not, but more so because outcome measures might be more robust to then ultimately make it approved quicker for all ages?
That's a tough one. It's very hard early in a technology platform in any particular disease to begin in any population other than adults. And it has to do with the regs, which is it's called the pediatric rule that if you enroll a child in a clinical trial, if there's more than minimal risk, which this would be, there have to be the potential for the child to benefit. Whereas in a clinical trial in adults, the benefit could be the information gained. So generally what the FDA will say, you come in for a request to begin in children. If you think that's appropriate for whatever reason, they'll say, no, you got to start in adults, and then there's a dialogue. In some diseases, SMA1, there aren't adults, but there's SMA3, which is sort of like that, in which there are adults. So that is the initial pushback. It's not as conservative in Europe about the opportunity to begin in children. Now, where you begin, this is part of a clinical development plan. So where you begin and where you end are maybe two different things, and you can even consider a strategy where you begin in one population that allows you to transition more quickly into another one. So the clinical development plan is probably one of the most complicated and would be the most contentious discussions the community is going to have sort of when you get there. But generally speaking, FDA, if there are adults, they're going to push for adults.
Could I just comment? if that were the case, and I have zero experience with the FDA and Jim has a lot, but I would push back very, very hard that what they're arguing for is not logical. You can do both, but I don't think that you have to spend 3 years testing adults before you can do the first child. I would feel that would be unconscionable and immoral.
But so we do. And let me tell you the most recent version. You propose children that the phase 1 study would be 3 at one dose, 3 at a middle, and 3 at a high, and then you go to 6. That's just generally how you start.
That also is a rule I would argue against.
No, I know, but I have to deal in the reality of interacting with the FDA, so one has to temper some of your thoughts about how illogical this may seem, but they do operate under regulations. But so the pushback was, no, we want to begin in children. So the negotiated position is the first one of the 3 is an adult, and the other 2 are children. so you know they're actually trying to accommodate you while quote unquote staying within the regs. So I do think there is an opportunity to and especially if the community, if there's a good scientific rationale and it makes sense from a clinical standpoint and the community speaks, then I think that's possible. But the other thing is once the platform is de-risked further, that would argue in favor of potential benefit for the individual. So we're trying to push the platform concept, and I think by the time we get there, I think there would be a good opportunity that you'd have flexibility in this regard.
And just to emphasize, none of this is to say that we're just pushing for kids to get it and not adults. The point is that you may see outcome measures that are more robust quicker in a young child than you may in an adult. Not that you won't see wonderful outcomes in an adult, but it may take a little longer. And if it takes longer, the trials may go longer, which means it will take longer for the whole community to get this as a potential FDA-approved drug or for a phase 3 clinical trial for all ages. So the goal is getting it as quickly as possible to everyone. It's not to choose babies versus adolescents versus adults. It's more of what can we do to get it to everybody as quickly as possible was the point of that question. This is for Dr. Beaudet. Do you feel any of the unsilencing strategies will give enough distribution and EB3A expression to have an impactful therapeutic benefit in humans?
Well, Ionis believes that they can get good distribution of their ASOs in primates by a lumbar puncture administration. I haven't looked at those data carefully, and I'm not familiar with the primary data. They feel that one can get very good distribution. But I guess my favorite approach isn't being pursued by anybody right now, but could be pursued easily, is to use gene therapy to turn on the paternal gene. And then so you get, to me, all the optimal biology, but you get a permanent effect. So if I was to do gene therapy, I wouldn't use the gene. I would use a strategy to turn on the paternal copy of the gene.
And then this goes to Dr. Wilson. Based on your experience with gene therapy in other conditions, what have you seen and what would you expect in terms of percent transfection and EB3A gene expression in the human brain with the viral vectors that you're using?
So the neurons in the hippocampus and cerebellum could be in excess of 50% or higher. The neurons in the cortex, we're really thinking more in the 10% range at this point.
Dr. Segal, can you explain the difference between an ASO and an ATF or zinc finger?
Yeah, so there are two strategies that are designed to turn on the paternal EB3A gene. And they just differ a little bit in how they do that. So perhaps everyone's familiar with this idea that we have DNA in our cells and that the DNA codes for RNA, and then the RNA codes for some kind of protein. Actually, in this case, there's an antisense that actually doesn't code for protein, so let's leave the protein part out for now. But let's say there's DNA that codes for RNA. And ultimately, what we would like to do to turn on this EB3A gene is to turn off this other RNA that's stopping it. And basically in the artificial transcription factor approach, we try to target the machinery that turns that RNA on in the first place. And in the case of the antisense oligonucleotide, it's a molecule that literally cuts that RNA so that it doesn't get to cause problems with the EB3A gene.
Thank you. This is another one for a few people on the panel. For each of your specific approaches, what is the biggest challenge each of you have to get ready for a phase 1 clinical trial? I think Dr. Wilson already answered that for us. Dr. Weeber and Dr. Nash? You kind of already answered that, but not exactly.
Yeah, I kind of answered that when on the talk. I mean, you have so many different combinations of these different things that you can put and package into this AAV. And then the question is, you know, the route of administration and where you target. So it's just figuring out the best combination of those things and the best route of administration to get the greatest effect. I mean, it's a huge challenge, and you have to rely on a lot of other people's work and a lot of other people's experience to try to narrow that down in a reasonable amount that you can put it into an animal model and actually get some results.
Dr. Segal?
Yeah, so I would say that our technology actually faces a number of challenges. That it's a little bit different than gene therapy. Gene therapy already faces a lot of challenge, but people like Dr. Wilson here have spent a long time trying to address those challenges, and so they're decades ahead. I think for the types of approaches like antisense oligos and artificial transcription factors, there's encouragement that there are some companies that are also trying to put those approaches forward. And in the case of SMA, we've already heard that there's an approach that's already passed through the FDA. So that's very encouraging for that approach. So you know that provides a road to follow for that. Although I'll let Dr. Beaudet speak to that more. For the ATFs, there have been clinical trials of zinc finger ATFs. It turned out that those ones were not as efficacious as they had hoped, but it does, you know, kind of chart a partial course for what might be required to get into phase 1 clinical trials. And so from where we are today, we would have to do the kinds of things that they did, show the kinds of efficacy studies preclinically that they did, show the kinds of safety studies that they did preclinically. But we'll definitely be looking at those as a model as we move forward.
Dr. Beaudet? I think you already said the humanized mouse. Okay. The next one, this is for Dr. Wilson. With a one-and-done strategy, theoretically, like viral delivery, will there be high and low doses? I think you just answered. The answer would probably be yes in the clinical trial. And if so, what options are there for participants if they get the low dose since they can't get it twice, or can they? If this is a spinal fluid delivery, isn't there immune privilege, in which case a second delivery may be an option? And if not, can you start with an AAV? And if you can't give that again, can you give another virus like a lentivirus?
The problem of rendering the patient immune against the therapy is a real problem that wears on me. But the strategy we've taken, which is directly injecting into the cerebrospinal fluid, we've dodged that bullet because you can re-administer through this CSF, because it's not really immune privilege other than the antibodies that interfere with your delivery are pumped out of the cerebrospinal fluid. So that you suggested in your question wouldn't be a problem with that approach. So you could use another AAV, the same AAV. But when you inject the vector into the blood, then the antibodies are very high, and then there's enough time to bind to the vector, and by the time they get to where they need to go, they've been neutralized. So you could go with an AAV or a lenti, using an intrathecal approach, or an AAV and an AAV.
Great. Okay, this is for Dr. Beaudet. Based on preclinical studies, what do you think is the most robust change we will see in humans after various gene editing or gene therapy strategies? And are you expecting drastic improvements or slow gains in replacing or activating the EB3A gene?
Well, I guess I'm a big fan of correcting. You showed some primary corrections and secondary corrections. Being a geneticist, I'm for fixing the genetic problem and not trying to work downstream of that. I think if you do that, in something like hemophilia, I think you should get a complete response at any age. But then if there are developmental things, there are windows that close where, actually, kind of thing of a good window that people might be familiar with, if you have a child with a crossed eye, you patch it and make the child look out of the less used eye so that they won't become blind in that eye. If they don't get the experience of vision through that eye by a given age, that eye will be permanently blind. And that's a very good example of a developmental window. And we don't know what parts of this story are limited by developmental windows and what part can be fixed at some future date and still get a very good result. So I think it's very hard to answer.
Great. Dr. Nash, can you explain where in the gene where the gene is going from the viral vector and where the viral vector comes from? Does the virus cut into the chromosome? And how does the virus and the gene then know where to go in the brain?
So you can imagine viruses that you get every day, like a common cold. Those are, if you imagine, they're like a sphere, okay? And inside they keep their DNA or their RNA safe. And in that sphere, it has particular proteins on it. And these proteins are what enable the virus to enter the cell. And those particular proteins are also which target the particular cell they're interested in. So for example, you don't want, if you're a virus, you don't want to be entering a liver cell if you really want to be in the lung. So the targeting of the virus is really dependent on the virus you use. And as Dr. Wilson talked about earlier, there was different serotypes of AAV, and some of those target different parts of the body a little bit better than others. So it's the sphere, the capsid protein, that really targets where the virus is going. So we choose which serotype we want to use so that we can get it to the brain where we need it. Now, once it gets there, it doesn't matter which serotype we have, they all deliver the DNA to the nucleus where it needs to be to produce the protein. So once it gets into the cell, it delivers the DNA, and then that DNA can make the RNA, which makes the protein that we're missing. And so it's more the virus that targets where it's going, but once the virus gets there, it kind of knows how to deliver the DNA. Now, with AAV, the DNA does not get spliced into the human chromosome. So what it does is it remains outside of the chromosome in what we call an episome. So it kind of forms, if you get multiple of these, it forms like a circle of DNA. And that circle of DNA can be maintained in the cell, and it can keep producing the protein that you need, but it's not actually integrated into the genome, which is actually very useful because, for example, if you have Lentivirus, it can integrate into the DNA, but we can't really control where it goes in the DNA. So if you can't control where it's being put, it can be put in the wrong place and cause another problem. So the advantage of AAV, it doesn't really integrate, so it remains outside of your own DNA, so it's not going to hopefully screw up anything else.
Thank you. This is for Dr. Segal. Are there any therapies in clinical trials currently for artificial transcription factor technology currently paving the way for Angelman syndrome with your technology?
So there was a artificial transcription factor that was put forward by Sangamo Biosciences. It targeted the VEGF gene. They were using it for a peripheral neuropathy, and basically what VEGF does is if you can get it to be expressed, it makes new blood vessels form. And they were injecting that into muscles first of animals, and then eventually it went into clinical trial. It made it through the phase 1, which meant that it was safe. Now this is the zinc finger, so it's the same basic kind of protein that we're using for our transcription factor. And so we're encouraged by that safety data. And then it went into phase 2 clinical trial. In phase 2 clinical trials, what you're looking to see is their efficacy. Is it actually doing the job that you want it to do in the person? And there they found that it wasn't doing the job as well as they had predicted from their animal models. So it stopped in phase 2. So that trial is now over, and they're not pursuing that anymore. They are pursuing gene editing tools, which are things that are designed to cut the DNA and then to insert other genes. And those are going into clinical trial right now, and it's still early days, but the first in-human nuclease trial with a zinc finger nuclease just occurred a few weeks ago. So we're anxious to see how that goes. That's not the approach exactly that we're taking, but those are the same basic types of molecules that we're using.
Wonderful. This is for Dr. Dindot. How come CRISPR-Cas9 has not been developed for a therapeutic for Angelman syndrome, and can you discuss some of the issues using this in human applications?
So I think that there are the intent to use it, but currently where that technology stands, it primarily relies on creating mutations in the DNA. And so as far as we know, that hasn't been explored as a way to, let's say, reactivate expression of the paternal allele. There is the new forms of that enzyme and that system that have recently come out to where they can target RNA, and so that's a possibility. But that technology as it's being used right now is primarily to create mutations in the DNA. And then there is some debate whether or not the technology is as accurate as we think it is, and so there's a chance that there could be mutations that are created in other places in the genome that are unwanted. But it's very promising technology, and I suspect it'll be, molded into something that we'll be using at some point, but not right now. Not my opinion.
Yeah, Dr. Beaudet.
So, I think that if you come back here in 2 years, there'll be 3 or 4 groups talking about using CRISPR. And one way to use CRISPR, we demonstrated some years ago now that if you put a stop signal in the antisense segment, you can stop the antisense there and then activate the paternal copy of the gene. So using a CRISPR strategy to insert a stop transcription signal in the right part of the antisense transcript would be a very attractive strategy, and I think it will be, as I say, 2 years from now, I think you'll be talking about the data.
Yeah, I would agree completely with that. I think at the time that Dr. Beaudet's landmark study showing that they could make a transgenic mouse and inserting that stop codon, at the time that came out, there wasn't an efficient way to do that in the brain. And I think that's been a real hang-up up to now. But there was just a paper, I think, that came out this week, if I'm not mistaken, where a new approach using CRISPR-Cas was showing that you could get these targeted insertions in brain at an efficacy of like 10 to 30%, which is much higher than we've seen in other places. We still have to see if there's good distribution of that and how you could replicate that, and there's a whole host of issues with that. But this is a very fast-moving field. So come back 2 years from now, yeah, you probably could see that.
We're not coming back in 2 years from now because this is going to be done. This is for Dr. Weeber. Are all the different treatment options permanent? And for those that aren't, can we look at that as a benefit?
Okay, so this is going to be one of those questions I'm going to dodge. I'm just going to tell you right now. Are they permanent? I think that what we've talked about with AAV, those, the AAV that you're putting in, it appears that it would be permanent, that you wouldn't have to undergo multiple treatments. I think, any other strategy that you're using, especially if you're using a strategy where you're coming up with a therapeutic to overcome the absence of maternal UBE3A, then it would be like any other drug that you take. So, so it would be something that you would have to probably take for the rest of your life.
Thank you. So at this stage, just to translate what that means to people, on the paternal, the strategies we're looking at for activating the paternal copy, which are ASOs and zinc fingers, or ATFs. ATFs, we're not sure the delivery method, so that could potentially be a permanent method, but if it's a viral vector type of delivery. But where it stands now is gene replacement therapy is permanent if it's associated with viral delivery, where activating the paternal at this point is not permanent, but that could be modified at some point to become permanent if it is seen when it's not permanent to be very effective. Is that correct?
Yes.
Thank you. I'm the translator. Okay. So this is for Dr. Wilson. What is the difference between adenovirus, adeno-associated virus, and lentivirus, and why are you choosing adeno-associated virus, except for the fact that you discovered it?
Well, I discovered the new ones, but not. So they're all viruses that are made, engineered to be made as vectors. Adenoviruses are common cause of sore throats when you're children, red eye. And they were developed quite aggressively in the 90s, a little bit beyond, like AAV for in vivo gene therapy. But they turned out to be very pro-inflammatory, so that there was a lot of immune toxicity. So they were shelved for that application. Adeno-associated virus is not related structurally or in any way to adenovirus, even though its name sounds the same. And as I described in my talk, that they're not very inflammatory. They do not very little immune toxicity, but they're primarily used to target cells that are not dividing, that are around for a long period of time, such as neurons and photoreceptors. Lentivirus is based on HIV, actually, but it's been engineered to be safe. But its application is primarily when you want to engineer stem cells or cells that are cultured, and then you want to transplant those cells. So really where the field has kind of emerged for viral vectors is AAV, injecting into people to target cells that don't divide, such as neurons, and then lenti, if you want to take cells out, such as the program you may be thinking about in a hematopoietic stem cell setting. Take cells out, the lenti puts the gene in, actually integrates into the chromosome, and then is transplanted back into the patient.
Great. And along those same lines, for Dr. Wilson, can you explain the potential risks and side effects of both gene therapy and the delivery methods that are possible for gene therapy?
For AAV?
For AAV.
Okay. There could be procedure-related risks. Whenever there's a procedure, there's potentially a risk of the injection. For AAV, the toxicity that we would be most concerned about is some sort of immune toxicity, despite the fact that I have argued that we do not see that. It's far less than in other viral vectors. So immune toxicity could be acute. When you inject the vector, you could get acute inflammation, or it could be a chronic inflammation. And what's really interesting about that is you never see it in mice, never, but you see it sometimes in monkeys. So mice are useful in some ways, or rats, like can you get the gene in and will you show function, but you really have to go to a different species, and especially primates, to look for safety considerations. Turns out that we've done so many different preclinical studies and so many monkeys. We have 200 monkeys under study at any one time. Really the only toxicity that we've seen so far, and we're starting to report this at high dose, whether it's systemic or not, is some toxicity in the sensory neurons. peripheral, that would lead to a peripheral neuropathy. And it's consistent, but only, and it's dose related. But we haven't seen any of the other more significant and worrisome toxicities, which could be inflammation of the brain. I mean, you just don't know until you do the experiments. We haven't seen that, unless you put a needle in to the tissue and give high dose factor, and then you can see it.
Thank you. This is for Dr. Weeber. Knowing our kids as well as you do and their sensitivities to illness, do you anticipate children with AS will have a higher chance of an adverse immune reaction with viral vectors than other individuals?
Short answer, no. I don't believe that.
That's good because I don't believe that either.
Yeah. So.
But I had to ask. These are not my questions.
It's less than my minute.
Dr. Dindot, have you tried any of these trials on pigs?
No, we haven't. So the Angelman pigs that we developed are at Texas A&M, and we're currently establishing the colony to use them in these studies. But we're waiting until we have sufficient animals to do that, and so none of them have been tested on that, or them.
Thank you. Actually, this is another one for Dr. Weeber. Sorry. Why does it take so long to do these preclinical studies and get them ready for clinical trials?
So I have
Hurry
to
up.
say, yeah, I'm going as fast as I can. I have
No,
to
I
say
meant
that
with the
working with Agilis and working with a company, man, it put a whole new perspective on doing research, because they are, they have deadlines, and they have really kept Dr. Nash's and my feet on the fire to get this done as quickly as possible. So we're doing it really as fast as we possibly can, and it is, I've really learned to whip my people into shape and get them to work faster. But it takes a while for those very reasons. I mean, after you get the virus and you've done this construct, and usually it's Kevin the one that's doing it, you get the virus, you have to inject it in, you have to wait 5 weeks, and then you have to do all these tests on them to make sure that what you're putting in is actually having an effect. And that takes time and energy and a lot of animals and a lot of personnel to do that, to run them through that water maze. It looks really cool. Yeah, stare at that for 8 hours a day for 3 weeks in a row. Yeah, my people go crazy doing that. So it takes time. It just really takes time. But we're going really as fast as we can.
Thanks. This is for Dr. Dindot. And this is from a parent who has 2 children with the same point mutation. Why are 2 boys in the same family with the same point mutation so different in their skill sets? Would we expect dramatic differences on the effect gene therapy would have on individual children missing the same gene?
So I may want Dr. Beaudet to answer this question, but I think my opinion, there are 2 opinions. One, in terms of the expressivity of symptoms, I think we've always thought that this was due to genetic background effect. So people are, different, for the genes that they have and so forth, and so that has an effect on that mutation and how it's expressed and so forth. I think maybe looking at some of the mouse data where we don't always see, for example, the epilepsy or the seizures, where you don't see all the mice exhibiting that, even though they're for the most part highly inbred. There may be other epigenetic factors that or things like epigenetics that play into that. As far as the treatment, I think that would be the amount that they get. I think that it'll be more appropriate to answer this, but I think there would be some variability in terms of where the virus goes and how much gets in and so forth. And so there would be some variability in terms of just the amount of EB3A that's being made, the number of neurons that are being transduced and so forth. But I don't know if anybody else has comments.
In a genetic situation like this, there are other genes and modifiers, is a term used sometimes, but typically the most potent modifier is the other copy of the gene. So you could imagine that paternal chromosomes are variably leaky. From some fathers, they express at a low level, from some fathers they express at a little higher level. So if these two children inherit different paternal chromosomes from their father, they may inherit one that's more leaky than the other, and so the actual amount of UBE3A available to the neurons in one child might be different than the other based on differences in the paternal contribution.
And along those same lines, do you feel that that might be a similar explanation for why some kids with a very large deletion have a higher skill set than some kids with a smaller deletion size?
Yeah, I would say if the single most credible speculation, okay, about the most likely variable when you see children with whatever genotype they have, that they vary, the most likely source of variability in my mind is how leaky the paternal chromosome is. And if you were fortunate enough to inherit a paternal chromosome which didn't make any antisense, you probably would never know the child had the Angelman mutation on the other chromosome.
Great. Another question, which I think might actually be great for Dr. Beaudet as well, and Dr. Dindot, is about overexpression of the UB3 gene. So there's obviously a lot of concern, and most of that concern seems to come from the industry of being worried that we're going to create a Dup15q model rather than human, rather than an Angelman human. And can you tell us what, if we say, you know, activation of the paternal copy may give us 80% expression of UBE3A, do you need 400% expression to have symptoms like a Dup15q, or do you need 105% expression to have overactive symptoms like a Dup15q? What is the real concern about creating an overexpression?
Well, there's a particular subset of the 15q dupes that are interstitial dupes, which have a more limited part of the genome. And many of them have 4 copies of UBE3A, 3 derived from maternal and 1 from paternal. And in those interstitial duplications, if you inherit the duplication from your mother, you typically have autism. And if you inherit the duplication from your father, you typically don't have autism. So it's not a cut and dried and totally convincing story, but there is some evidence that overexpression of UBE3A could cause autism-like findings. If you do something like gene therapy, you're going to have some cells that don't get any expression and some cells that get higher expression and lower and so on. So that's really the main reason I made the comment that I think if I was going to do gene therapy, I would want to inactivate the antisense and activate the paternal copy of the chromosome, because at least we think everything will be turned on at the right levels when we do that. And we don't have to worry about overexpression if we knock out the antisense and activate the paternal copy of the gene.
And what if it's a UPD child, who then you're activating 2 copies of the paternal?
Yeah, on the one hand, they might be especially prone to benefit if your treatment is suboptimal, but if your complete treatment is completely optimal, they might have some harm. So that's a very interesting and tricky question.
I will say that this is something that we're looking at. So, you know, getting good at getting a large amount of expression of UBE3A in the brain also allows us to do this in a typical rat or mouse that already has 100% of the UBE3A there. So we can give the virus to that animal and see if that increase in the amount above 100% changes its symptoms or changes its phenotype at all. So yeah, I think that's something that in order to go forward with this, we need to explore, but there's ways to explore that with the models that we have now.
Wonderful. This question is for Dr. Wilson. In your opinion, and you kind of dodged this before so that you can answer it here, in your opinion, if these strategies are successful, do you anticipate they will be affordable and accessible to all individuals with Angelman syndrome, national, international, insurance, no insurance, et cetera?
Well, you know, internationally, even current standard of care isn't available to most patients. Most patients with genetic diseases don't get current standard of care, if you look at India, China, and others. So that's a larger issue. That, so it would be affordable. You know, this whole business about how to price the cure and whatever, we've been talking about it, and now that it's real, there is a lot of effort. In fact, the government just weighed in on it today to develop some some laws around pricing structures. So, I think as a society we're going to figure it out if these are really transformative therapies. I know that doesn't help, but the other point, as I've been at Penn, it's hard for me not to visit with my colleagues at the Wharton School to ask them this question. I've been asking it for 25 years. But the one question that I was asked when I asked the question is, what's the cost of development? Well, the cost of development, let's say an SMA trial, it's not that much. The clinical trial is going to be, the pivotal trial could be 18. The actual cost of development of that particular product that could weigh in terms of justifying the price is low. It's really what will the market bear, other pricing structures. So now, and then you have to weigh in failed programs and whatever. But what most of these payers are moving toward is not a single high payment. It's an annuity-based system. That's something we've been talking about for a while. So if you get your treatment and it works, then the payer pays out over time as long as it continues to work. But again, if this is really helping people, we're going to figure this out. We just have to get to the point. There's a little bit of a catch-22, but and I've been involved in biotech companies in the ultra-orphan space, and there is no business model that makes any sense that justifies their valuation right now. But they're valuable. I don't know what the hell that means. But I called it the land of Oz.
Dr. Beaudet, go ahead, and then I have a question for you.
Well, there are a couple of examples where we've seen, I think if there are really transformative therapies, they will be available to people, and maybe the HIV drugs may be an example, that they're reasonably available even in very underdeveloped parts of the world. I mean, so I think if they're really transformative things, and when renal dialysis means that either you die or you keep getting dialyzed, at least in the U.S., there's been a funding mechanism for dialysis that's been generated. I don't know what that's like in underdeveloped parts of the world, but I would be confident that if it's good enough, I think, as Jim says, society will find a way. We'll figure it out.
And along those lines, can anyone, maybe Scott or Dave or Art, can answer on the panel, do you have any understanding or ideas that we can kind of feed off of from the SMA approval of Spinraza, where they have been approved? It was an expensive trial, and it was expensive development, and the fact that regardless of that and its cost, what the insurance companies are doing with that, so that now it's an approved drug, is it being available to all children, at least in the United States, that we can speak for?
I don't know the status of payments for SMA.
I do, fairly. And I mean, what was amazing is how quickly the FDA got behind this product based on really outstanding data. Caught the company off guard, quite frankly, because they hadn't done all the groundwork from a marketing and commercial standpoint. And at the end of the year, they had a broad label beyond what they had data for in terms of patient ages. What's interesting is FDA approval does not, it's not used by payers to determine payment. So for adults, for example, with SMA, the payers are asking for evidence that it's working after the loading dose. So you've got to develop a data set that would convince the payers. But Biogen has a policy that if you try 3 times and the payer says no, then they'll pay for it. That'll pay for the drug, but there are other associated costs with injecting something, every 4 months and whatever. So that's pretty much where it is right now.
And I would just say, I mean, this may seem kind of obvious, but these are early days, right? These are early days of really, technological advances. And so, we didn't start out with this, right? I mean, first computers were gigantic. They were very expensive. Only governments could pay for them. Then big companies could pay for them. Now everyone can pay for this, right? And there's many examples of that. These are early days. This is foundational things that are happening, and they're just really happening now.
Okay, well, we have time for questions from the floor. I think probably most of your questions have been answered. That was the goal. But are there any other questions from the floor? Yeah? Runners want to grab the mics?
For Dr. Dindot, I think you said the pigs would be ready later this year. And where are they going to go? What are they going to be used? Which modality? All of them?
So yeah, if the breeding schedule stays on track, then we should have them, here within the next 8 to 10 months, and we're building up enough where, we should have a large enough number of them to start working with. Where it'll start, though, is we'll first characterize the model. And so, there's, we know a lot about the phenotypes in both the patients and in the rodent models, and we suspect that they'll be, very similar. But, these are much larger animal. They're much more sophisticated. They're very intelligent. And so, and there's really no precedent for this. To my knowledge, there's no large animal genetically engineered model of a human neurogenetic disorder. I mean, there are for Alzheimer's, but not for developmental disorder. So I think, we'll spend quite a bit of time trying to characterizing the animals and seeing, just what they have. And then, as quickly as possible, the plan has always been to move these therapies into them, to have a sort of a 2 or 3 tiered system where things are tested in the mouse, perhaps, the rat, and then, tested in the pig. And so the design was not to have them being used as an experimental model, just kind of like we use mice, but rather a sort of a bona fide preclinical model for therapeutic testing. But, we'll go in stages and then, move as quickly as we can whenever, we do that.
Question here for Drs. Wilson and Beaudet. If you're turning on the paternal allele and in a sense turning it into a maternal allele that works, would you have a risk of turning an Angelman child into, say, a Prader-Willi child who has a missing paternal allele but a working maternal allele?
Depends if you do it right or if you do it wrong. If you knock out the antisense up by its promoter, that would create an Angelman partial phenotype probably, because certain of the SNO RNAs have to be expressed and they would be lost if you did it upstream. This antisense is a very long transcript. If you do it downstream near the Angelman gene, you leave all the SNO RNA expression intact and you can still activate the paternal copy of the gene. So in a way, I've seen that as it's so fortuitous. I mean, if the antisense were in some other relationship to some of these other genes, you wouldn't be able to knock it out and leave the Prader-Willi elements intact. So if it's done in the right location, it should be fine.
A lot of our kids express the Prader-Willi excessive hunger. Is there any risk that once you remove the antisense that there's a possibility that there's a portion of the paternal missing as well that was missed in our original diagnosis?
No, I think that's the same as the last question, basically. If you can, it's just fortuitous that the antisense is at the end of this very long transcript. You can knock out other upstream parts of that very long transcript are necessary to prevent Prader-Willi syndrome. But it's possible to leave all that intact and knock out the antisense. And so it's actually a pretty interesting thing that you can do something that nature hasn't evolved to do, is to take one chromosome and make it do for both Prader-Willi and Angelman.
I guess my question is, if the the chromosome already doesn't have the ability to produce UPE3, and we're relying on that chromosome to produce it by removing the antisense, is there any risk that it won't be able to do that?
There's a fair amount of mouse data that says that it works pretty well. And when people have tried to look at the start of transcription from the paternal and the maternal copy of the gene, it's interesting. I was rather shocked to learn when we did that the paternal copy of the gene transcription is being started, it's going through the first few exons, and then it's hitting a wreck halfway through the gene. So it seems like the paternal and the maternal are similarly regulated, and it seems like removing the antisense, I think to the extent you can completely remove the antisense, if you blunt it but don't completely remove it, then the expression from the paternal chromosome will probably be insufficient. But there's a lot of speculation, but I think it seems like everything is aligned in a very fortuitous way.
There's a lot of hands. Any over here?
Nelson over here.
Over here.
I have two unrelated questions, but I want to just get them in here. Are there any other disorders where one gene is silenced that we know of? That's the first question. The second one is, are there any naturally occurring viruses that attack the brain?
Go ahead. Ashley.
If I understand your question correctly, you're saying is there a similar neurodevelopmental disorder where there's one copy that's silenced or missing? So actually, we're working with 3 neurodevelopmental disorders right now. Angelman's one of them that have 2 flip sides. So where you have a concern on a duplication for gene therapy, that becomes one of our concerns. And for one of them, we have a similar situation, not exactly the same, but it's an X-linked disorder. So most of the time, at least in this pediatric epilepsy, it manifests in girls. Most of the time, boys don't actually develop the disorder unless they're what we call mosaic, where only a few number of cells end up expressing the wild type. So what we've talked about in that disorder as a potential therapy beyond gene therapy is, can you unsilence in the boys, in the boys you have a Y chromosome that doesn't carry the allele, and in the girls you have 2 chromosomes, and normal X inactivation happens in the female brain where 50% of the cells express that mutated form and the other 50% express the wild type. So if we can unsilence that silenced wild type, we call it X reactivation. It's a potential therapeutic hypothesis, I would say, at best at this point. So similar, not exactly the same process, but again, talking about unsilencing something that's normally silent. So not really what we're talking about in the context of gene therapy, but when you're talking about the imprinting and really tackling the paternal allele, it becomes similar discussion, similar concerns, and what the effect would look like. Does that answer your question?
Yeah, I was just wondering if you could solve, I guess ultimately if you could solve the problem, would the technique have applicability to other diseases that may have a situation where one of the alleles is expressing and the other one is
There's a handful of such diseases, but we're talking half a dozen or so, and actually maybe the best example is Prader-Willi, except in that case it's maybe some combination of genes in the Prader-Willi region rather than a single gene. And certainly in the deletion Angelman patient, it's not just the Angelman gene either. And I think the suspicion is that if you corrected the EB3A expression in a deletion patient, that this would solve the bulk of the problem, but there still would be some other genetic differences in a deletion patient.
So your other question was viruses that go to the brain? Yeah, there's been a lot of work over the years using herpes simplex virus, which does take up residence in ganglion and causes encephalitis. But there's no advantage in that. The way that we attenuate these viruses, it's a one hit. So whatever cell interacts with the virus or the vector is the only cell that takes the gene up. No one has yet successfully developed a system where you could seed a tissue and then it would spread throughout the tissue. That would be sort of the holy grail. But the problem is, in the context of doing that, it elicits pathology in their immune responses. So there's really no advantage at this point.
Could I mention just one other advantage of Angelman and the way this situation is? If we're talking hemophilia, you have to worry about immune responses to the factor VIII protein. In Angelman, we don't have to worry about immune responses to UBE3A because UBE3A is already being expressed in the liver and kidney and lung and whatever from the paternal chromosome. And so while there's been some talk about immune responses to the vectors, and that is still a factor, there are many things that are just very fortuitous about the Angelman syndrome that make it a very favorable situation.
You had a question?
My question is pretty simple. There is not currently an Angelman primate. Will there be one?
I think yes. I think it's technically possible to make a non-human primate model of Angelman syndrome. I think the question is if the costs and the benefit of that model, or if the benefits of having that kind of large animal model would justify the cost and maintenance. And would there be questions that could be answered there that couldn't be answered in, say, the pig model?
Yeah, I think you think research is slow. Try using primates, where you have to breed to get a whole cohort of Angelman monkeys.
That would not be a pretty monkey.
Yeah, and I mean, I'll comment on that. There's
we all live with a little monkey.
the age at which they reach, so they're singletons, so there'll be one animal, one offspring that's born. The age to sexual maturity, so it could be years, rather than months. And I think the ethical issues is probably the biggest thing, and maybe not so much the ethics of making one, but just how the public perceives it at these institutions. So there's often a lot of pushback for using animal models, and that's one that's very polarizing, amongst most people.
Somebody is going to be the first human patient, and somebody is.
Yeah, and I mean they've made
We're not fighting for that spot.
no.
Speak for yourself. It might be.
I didn't know if you'd get more data from a primate of some sort before it was one of our kids' brains with the injection.
Could I just maybe comment on the large animal model? So I was involved heavily in cystic fibrosis research, and the mouse didn't cough, so that wasn't good. But there were some features of the mouse that one could use to try to evaluate treatments. And then Mike Welch from University of Iowa proposed to make a pig. Now, the pig got very sick and was sort of a tedious model. But I'm just sharing with you the response of that to the community. There was concern from the community that the prospect of a pig could actually slow down and deter the development of novel therapeutics. And why? Because it takes a while to breed pigs. They weren't that accessible. And if you have to get in line, and that's a gate to moving something into the clinic, just be prepared for timelines that are going to be extended significantly. I'm not saying that you shouldn't do it, but I'm just saying with CF, they were really worried that that became a gate. And I was sitting on NIH study section. It became a gate. It became what everyone should use. I can tell you, I don't think FDA would view it that way necessarily. And CF went forward, and they used an in vitro model for screening for molecules, and didn't go through a pig. And there was some success. So I think in terms of defining pathogenesis, it could be enormously helpful. But I just be careful about it becoming a functional gate for everything that has to go into a human.
And if I could just clarify also, the issue is not whether we want to use a large animal model or do large animal studies. The issue is whether we would want to make a non-human primate model of Angelman syndrome. So I think you could use wild-type monkeys, for example, to look at safety. So you could test the therapy in a non-human primate that was not an Angelman model, but it was wild-type, and you could get that kind of safety data. And I think in some cases you could also look to see, since the genetic sequences are similar, in some cases you could even see if the kind of molecular effects were also happening, if things were coming on and turning off the way that you expect. So you could get those kinds of things maybe even from naturally occurring monkeys in colonies without having to make an Angelman model. I think that most of the people up here would be planning to use a large animal model for those kinds of studies anyway.
Yeah, just to add to that and to speak kind of to the concern as being parents, a lot of what we've thought through with our program and a lot of what not specific to this program, is toxicity. It's a constant concern. And as I mentioned before, we're working in 3 neurodevelopmental disorders where that duplication flip side becomes a concern. We've thought a lot about what that conversation is going to look like with the FDA. We've talked to Scott about the pigs, looking at timelines, and ideally we'd like to move very quickly. I think the pigs become a real important option if there's concerns that end up coming to fruition. We will move through primates, and that ends up addressing the concerns about that duplication, because you are expressing a vector on top of wild type levels. So if you have a concern about a dup15q phenotype, that's where you'd potentially see it. So if we did find anything at that point that was a concern, that becomes an important fallback. But I think consideration of timelines, we do want to keep moving forward quickly.
Great, so we're gonna wrap it up there. We have 1 more minute. So in 10 seconds each, I just wanna let the panel have one last thing to say to the community. We're just so grateful that you guys are all here and the optimism and excitement you've brought to the community over the last 10 years, but I would say certainly in the last 3 to 5 years has been pretty phenomenal. So can each of you just have one last comment to the community before we close it up for the day?
I just want to tell you guys thank you for your support, for being here year after year, for sending me emails and telling me to keep going. It is really a privilege to be able to work for you.
Yeah, the same thing for me. I'd like to say thank you. You certainly give us motivation to keep working, and your enthusiasm keeps us going.
Yeah, I think the same. I think it's a very unique opportunity, and it's very motivating, and it's very rewarding. So thank you.
I think that a lot of people sitting in the audience here feel like you've come to hear what we had to say and that you want to have some answers, and you look to us for answers, and you think that that's the relationship. And I would just suggest to you, you don't really know your real power, okay? I mean, the FAST organization essentially is all of this. It's all of us, right? It's all of these people in this room. And, all the contributions that you've made, all the interest that you showed when you talk to the scientists, that's very inspirational. That helps the work go better. When you go into the registry and put information in there, and we could use that in studies, that helps things go better. I mean, a lot of what we can do, we can only do because of you. So really, I think it should be us that's thanking you.
Yeah. I think I've already said that the stars are all aligned in favor of Angelman syndrome being amenable to a really curative kind of treatment. And the other thing I would say is I've been very little involved with FAST up until recently, and I think FAST has really transformed the landscape of Angelman research. I mean, the Angelman Syndrome Foundation is out there. They've done a lot of good things, and a lot of us have been involved with them for many years, but FAST has really accelerated things.
So I'm going to make a plug for ABOM and for parent and child participation in clinical studies that precede when we get to the clinic. Participation in registries, which Megan is spearheading, and the Australian team, is invaluable. We can get the therapy started, but unless we understand the disease, unless we understand what your children are going through, unless we can figure out where in the world you live very quickly, this becomes a lot slower of a process. So continue to participate. I know it's time consuming, I know it's hard, but the more biomarkers we have, the better understanding we have of this disease, the better we'll understand if these therapeutics matter when they get to the clinic.
Well, other than thank you for welcoming Penn into your family, we will make sure everyone is relentless in moving forward without delays and interference. But the one thing that we need to be aware of is the science that's going to emerge. And I can promise you we will be transparent with that science, and I'm sure my academic colleagues would be. That isn't always the case from companies. I'm involved in many companies, but we're academics, and I think we need to pledge to you total transparency in the science, and we will have to pivot. We will have to pivot, but if we multiplex, life cycle management, stay determined, but the science is going to speak, and overall the potential is so huge, but we're going to be taking a few detours along the way, and just hang in there with us.
Well, thank you to the lone soldiers who remain in the room. I'm not surprised by any of the faces that are still here after this very long day, because we all know each other and we know those people that are never going to give up, and that's all of you in this room. So we're going to adjourn for today. Thank you all for being here. I don't know about you, but it was certainly an inspirational day for me, so I hope you feel the same. We have a really exciting day tomorrow as well, the Educational Summit, which starts at 9 a.m. And it is not in this room, it is in the Hyatt, is that correct? Yes. So you don't have to get lost. It's going to be in the hotel that we're staying at tomorrow at 9 a.m. And that doesn't last the entire day, but there are 2 separate rooms. So make sure you look at the agendas and make sure you decide which room you want to go into. One geared a little more toward anxiety and one geared a little more toward AAC and early access. And there's also the carnival, the AAC carnival, so that's here today and it'll be there tomorrow as well, so please stop by the vendors. And I really want to thank our sponsors. We could never make this happen, and all of us couldn't be here if it weren't for the tremendous amount of sponsorship we got to pay for many of our tickets for the parents to get caregivers at home to be with their children so that they can actually do everything they can in their power to help their children. So thank you all for coming, and thank you to our sponsors.